Hello and welcome to the EMJ podcast with me, your host, Dr. Jonathan Sackia. Today, I'm thrilled to be joined by Dr. Mark Connors, a senior investigator and chief of the HIV-Specific Immunity Section at the National Institutes of Health, the NIH. Dr. Connors has an impressive background. He earned his MD from Temple University in Philadelphia before training in pediatrics at Tufts New England Medical Center and infectious diseases at the National Institutes of Health and the Children's Hospital of Philadelphia before he went on to join the NIH. Since then, he's dedicated his career to understanding immune responses in HIV, with his research making significant contributions to the field. Mark leads pioneering studies in HIV immunology, and his groundbreaking work has been widely published, advancing our understanding of immune dynamics and helping shape new approaches for HIV treatment. Mark tells me that he's a very keen cook and an enophile. So I think we're going to start off with a question about that. We're truly honored to have him here on the podcast today. Welcome, Dr. Mark Connors. Lovely to have you.
Thanks, Jonathan. It's my great pleasure to join you today.
So I'm also, I love wine. I love good food. I'm actually, as I was telling you, I'm cooking for friends tonight. Actually cooking with friends tonight. If you were to design your perfect menu, what would it be, and what would your 3 favorite wines be? Whatever.
Oh, perfect menu is just so hard. If it's the last meal kind of thing, there's certain things that have like sentimental value to me. My mother's side of the family is Italian, and we grew up with fresh pasta, and having one of those meals is just, really grabs my heart. And so with that, I love to have beautiful Barolo or possibly something from Tuscany with plenty of age on it. And that, for me, is a perfect meal. I don't need 10 courses. That's satisfaction for me.
I went with a group of friends a few years back to the Barolo. Purportedly, we went there to watch a football match at Juventus. But that was 90 minutes. The 4 other days were spent going to all those amazing vineyards and tasting some great wines and eating some utterly fantastic. You know something? There are some great French wines, but there are no bad Italian wines, apart from, in my opinion.
Yeah, I have the same feeling about that area. We went in around 2000, so it was not yet touristed, and you could go to those wineries and meet the person whose name was on the label, and then the incredible meals out at some of those restaurants, and great hospitality, spectacular scenery, just really beautiful.
Yeah, well, I think we could probably talk about food and wine and travel for a long time, but I think we're supposed to discuss HIV. So can you tell us what first drew you to immunology and specifically to HIV research?
Well, I started as a fellow at NIH, working for 2 people, Robert Chanik and Brian Murphy, and they were really giants in the field of vaccines. And at that time, there were really the techniques for understanding immunity were fairly rudimentary. I mean, I was drawn by the incredible power of vaccines that I could see in animal models, and ultimately that lab had developed a lot of immunotherapies, the first RSV antibodies, and had discovered lots of viruses. So, and the power, of course, was put to me by Bob Janik, who said to me, you can go and be a clinician and maybe save 1 or 2 lives in your lifetime, but you develop a vaccine and you potentially can save hundreds of thousands or millions. And I'm not the only person he told that to, and I'm also not the only person who found that very powerful. And so I really kind of committed my career to that. But also what was exciting in the early 90s is that a lot of new techniques were coming on board to understand the immune response. And so we were understanding it then in a level of detail that had not been possible before when you had these sort of rudimentary assays. And really, that was what really drew me in and got me started. So I needed to make a decision, and it was to devote more of my time to research and less toward sort of clinical endeavors.
Yeah. to be involved at an early stage, an inflection point in medicine, is immensely gratifying because it's the great unknown, and there's so much work to be done that there's a rich vein you can tap, if you'll forgive the the analogy. But when I did my final exams many years ago, there was a question in my final exams, in one of the oral exams, about HIV/AIDS. And thank goodness I'd read the newspaper the day before, because that was all I knew about it. I knew nothing about it. So, and I was around when HIV first came to light. There was intense concern. There was even hysteria. And I trained as a surgeon, and we were all petrified about, what should we do? And how do we, obviously we have to treat patients, but how do we do it? How has the disease and our reaction to it changed over the years? I know obviously there's much more acceptance among the general public. And I always remember Princess Diana hugging an HIV/AIDS patient on one of her visits to a hospital. So talk to us about the HIV journey.
Well, as you said, it was a scary time in the early 80s. Like many physicians, I have my story of I was a second year medical student interviewing a person who had a lymphadenopathy syndrome, and I had to be gowned and masked and goggles and hairnet, all this stuff. And I'm interviewing this young man who had engaged in risky behavior and had acquired HIV, which we didn't know what the virus even was then. So yeah, there was a lot of first fear, and then there was stigma, in the field. People who were homosexual and that were using intravenous drugs were particularly affected, and so that whole period maybe delayed a lot of research. And then now there is much more acceptance, and the field works in partnership with advocates that are lay people out in the community, and so which is wonderful. So it has evolved tremendously over the years, but it's not perfect. We're still not completely there to where it's treated like any other virus, but it is tremendously better.
So your research is focused heavily on T cell responses in HIV, and you recently published a paper on this. Can you explain how those responses play a role in controlling HIV and why they're so crucial?
Yeah. So just for the lay audience, the way that we control virus infections is through killer CD8 T cells for the most part. And this is a system that we've evolved to deal with intracellular infections. So what the body does, what the cells do, is cleave up a virus into short peptides and put those short peptides on the surface. Your killer CD8 T cells recognize those peptides in the context of what's called MHC, compatibility complex proteins, and it recognizes that as not self and also recognizes it as dangerous and then kills that cell. And that is a very, very efficient system. Unfortunately, various viruses have many ways, and also cancer, have ways of subverting that response. And so we only see through evolution and coevolution of viruses in humans what's been successful. And in the case of HIV, it has devised a means of sort of shutting down that CD8 T cell response at the level of accumulation of proteins that kill. And so the infected cell is recognized by the CD8 T cell. It just, in most progressors, it just doesn't, it's unable to kill because you could think of it as those cells don't have enough bullets. They don't have a molecule called perforin that pokes holes in the target cell and then kills it. And so that by far is the biggest difference between people who don't control and people that do. So we have gone in. I learned this during my fellowship that it's very powerful to look at examples in nature where you have, for example, in this case, control of a virus, and then other examples of a lack of control, because it gives you something to shoot against. You can take that apart, right? And so we've done that for many years in an algorithmic way, and that is the final answer, is that progressors don't accumulate perforin, and that's why they don't kill. They recognize the virus perfectly fine, but they don't kill infected cells. The paper that we just published related to vaccines, we wanted to always apply that as lessons to vaccines. And so when we did that, we found a different mechanism by which vaccines were not working. So several very high profile vaccines that had failed to induce an effective CD8 response. What they did was they weren't pushing the response far enough. So we need more immunogenic vaccines, more shots to be able to push that response so that those cells are sensitive enough to see the low amounts of antigen on an infected cell and kill it. And I think that can be done. We've already started a lot of work in macaques to try and see what platforms are able to push that type of response. But at a higher level, I think that it's going to be very unlikely, at least from the perspective right now, that an HIV vaccine is going to be able to do what it needs to do without a CD8 response. You're going to need an antibody response for sure, but achieving something that's able to protect against so many variants of HIV that are out there and not just immediately select for resistance is a really, really tough thing to do. And whereas the CD8 T cell response would be a backup that would stop a person who does get infected, it would stop them potentially from transmitting. So for those reasons, it's very important that we push on and understand what the rules of the game are for generating an effective CD8 T cell response.
Fascinating, Mark. And I was doing a podcast last week with an old friend who's an infectious diseases doctor down in Florida. And one of the things we invariably spoke about was vaccine hesitancy. I have to say that name makes me twitch uncontrollably. It's sort of like, in the gangster movies, they never talk about shooting someone in the head. They talk about retiring them. They're going to go and sleep with the fishes. They're going to pop them a cap. You just did a wonderful job explaining vaccines in this circumstance. And one of the arguments I've seen from the naysayers about COVID, for instance, is that there's proof that vaccines don't work. There's proof that vaccines are just a way for pharmaceutical companies and doctors to make money, which is, of course, total nonsense. And one of the arguments that this lobby uses is that if it worked, don't you think they'd have made a vaccine for HIV? So my aside to you, this is a comment, not a question. My aside to you is you need to get out there and give a rational explanation as to why it is difficult and why so much time and money has been put into this research. So that was a personal thank you for explaining science that a simple surgeon like even a simple surgeon like me could understand. So thank you for that. So I want to go a little bit into the history of HIV. The first known case, I believe, was from 1959. There was a blood sample from a patient in Kinshasa in the Democratic Republic of Congo. But there might have been earlier undocumented cases where someone died of a strange scenario and it wasn't recognized and no blood sample was kept. But since 1959 and then the early 80s, the understanding of HIV has evolved greatly. What would you say are some of the most significant insights and inflection points about HIV immunity that have come to light in recent years? I'm sorry for the lengthy question.
Yeah. So one of the things that happened in the late 90s was that we had identified, let's talk about the T cell response, then we'll talk about the antibody response. But we had recognized that there were a rare group of people that are now called non-progressive elite controllers that could control HIV to below the levels that we detect in most of our assays. So just extraordinary control. And that really opened the door to the whole research that I've engaged in the past 30 plus years, where you could take that apart. And the answer to how those people do it is actually quite extraordinary. There were many false signals in the late 90s, early 2000s, and a lot of those were attractive ideas. Okay, so the first thing was, oh, well, the people that are progressives just don't have enough CD8 T cells. Nothing could be further from the truth. Everybody walking around with HIV had 20 to 40% of their peripheral blood CD8 T cells were devoted to HIV. That's an extraordinary number. And eventually that got accepted as fact. Then there was another attractive idea that the virus escapes your immune response by mutating, and that's how it's beating it. And that took a very long time to undo, because it's a very attractive idea. And if you go in and you sequence HIV, which is very variable, you're going to find mutations. You just do. But there's never a difference between people that control and don't control. So there were all these observations that happened over the past 20 years that were sort of landmarks in our understanding of the CD8 T cell response. I think that most people agree now that, actually more than most, that almost everyone agrees that there's no correlation between how many cells you have and your amount of virus you have in your blood. There's the virus, the autologous virus is recognized perfectly fine. And so we can move on from there, and we know what we need to stimulate in a vaccine. I think that those were the three things that really needed to happen for the CD8 T cell response, and we can, as a field, move on now. The B cell response is equally interesting. In the early 2000s, we had recognized that there were people out there whose antibodies could neutralize in vitro many different isolates. And the reason that was incredibly important was that by far the most important obstacle to HIV vaccine was the incredible diversity of HIV. So for example, there's more diversity in a single person in envelope than there is an entire epidemic of influenza at the level of hemagglutinin. So it is quite extraordinary that these people were able to recognize and neutralize viruses that they had never seen before. And so our group and John Muscol, WRC, Dennis Burton at Scripps, Michelle Nussenzweig at in New York, all of us had begun isolating these antibodies, and several other people I'm not mentioning had all contributed to this work. And the answer to how those people neutralize HIV, their antibody response, turned out that it wasn't because they had lots of different antibodies to lots of different places. It's actually that they had a few antibodies to about 5 different conserved patches on HIV envelope protein. So they were doing it by targeting these conserved patches. And so we all isolated these antibodies. Those antibodies are now being used by big pharma as immunotherapies, and it's also been used as prophylaxis in trials in Africa, for example, and had given us a proof of concept of what we need for an antibody response in a vaccine. Unfortunately, the bar for that is going to be very high, but we do have something to shoot against now in terms of how much we need and what specificities that we need. So I would say that those are the major sort of obstacles that have been overcome. And just one last bit of perspective here, Jonathan. I was Tony Fauci had said to me quite a few years ago, pre-COVID, that he didn't understand that a lot of people didn't understand the importance of an HIV vaccine at this point in the pandemic. And I said, well, it's the moonshot. It is the most difficult virus out there to generate an immune response against, for the reasons that I just laid out to you. And so it generates all these technologies like antibody isolation and our understanding of the CD8 T cell response, and all of this technology has come. And then sure enough, probably about 5 years later, COVID came, and all of those guns that you saw turned on COVID, nearly 100% of those came from the HIV field. We just took that stuff, all those vaccine platforms, and turned it on COVID. But those were decades of research that preceded COVID that largely came from the HIV field. And so that's the importance of doing this kind of work on hard viruses, is that you will have a better leg up on all viruses.
Yeah. Going back to my prior point, Mark, I think that we did, as a profession, we did a crappy job telling people, because the naysayers have got it in their head that these vaccines were dreamed up with no research and no thought, and that it was, a couple of weeks work, stick some carbon nano. That was one of the things: carbon nanotubules and GPS location devices, God knows, into vaccines. But we, as a profession, need to do a better job. But, I was always telling people, no, we've been looking at this stuff for many, many years. So just as I was this ignoramus going into my finals, thankful that I'd read a newspaper article, if there was a medical student today who came to you and said, what do I need to know that's, new and exciting and challenging in HIV, what is it? What would you tell that med student?
Well, I mean, I think we're about to see another inflection point in HIV vaccines, and also an inflection point in the sort of merging of our knowledge about how to deal with chronic infections like HIV and CMV, and merging that information with cancer immunotherapies. There's tremendous convergence of those fields in our understanding of how cancers beat the immune system and how chronic viral infections beat the immune system. And the reason I think that there's an inflection point is some newer tools have come online that are really allowing us to get a much more detailed picture. I mean, beyond the sequencing of the human genome, which tremendously helped, we now have all of these single cell technologies where you can look at the transcriptome, so all of the RNA transcripts in a single cell, and look at individual subpopulations within the CD8 T cells, for example, that react to HIV, and see what's similar and different between those subsets. We also have mass spec proteomics, where we can just look at the proteome of sorted cells. And so that proteome allows us to look at all the proteins that are being expressed by a cell. And so I really can foresee in the next 10 to 20 years just an explosion of knowledge about how this all works. And, instead of just counting cells and saying, oh, they make this, they make a little bit of that, we're going to get a very, very detailed map of how this all works. And it's a tremendously exciting time for both infectious diseases and vaccines, and for, I think, cancer immunotherapies.
Thank you for that. So we've talked a little bit about vaccines, and I noticed that Dr. Huang from your lab is lead investigator, looking at a growing consensus that, we've talked, as I say, about neutralizing antibodies and potent T cell response. You've said how important that it will be for an effective HIV vaccine, and it's been challenging. Can you share a little bit more? Can you expand about the challenges and the breakthroughs in developing an HIV vaccine, and how your lab's work is contributing to it?
Yeah. So for the T cell response, the challenge right now, based upon what we've just published and what we're observing currently in experimental animal models, is that, and this actually has a lot of crossover with the B cell response, and I'll explain why in a second, is that you need persistence of antigen. You either need to give a lot of shots, or you're going to need something that just stays in the body for a longer period of time and replicates. So the only examples that we've observed so far, either in humans or animals, and when I say we, I'm talking about our laboratory, because it's a relatively new thing that we published last December. The only time we've seen cells with sufficient sensitivity is when you give a human, for example, vesicular stomatitis virus vector after they've received several DNA shots. So they got a lot of shots and a lot of antigen. We see high sensitivity responses in chronically infected, elite controllers. We see high sensitivity responses in elite controller rhesus macaques, for example. So the challenge there is going to be persistence of antigen, or giving a lot of shots. The people need to understand that as quickly as possible and get on it. Some of those vaccines, unfortunately, have been abandoned by companies, and they probably need to be resurrected and re-examined. On the B cell side, on the antibody side, it's a similar situation in that you're not going to get away with an HIV vaccine where you have this up down pattern of neutralizing antibodies similar to what we see in COVID. For influenza, we get away with that because we need a new vaccine every year. The virus changes sufficiently that you're going to need a new vaccine every year. But for HIV, that's not what you want. You don't want to be re-immunizing all of Africa every 4 to 6 months. You need something that's going to persist, and that's the way that you get durability of the antibody response. A lot of people don't understand that a lot of those live virus vaccines that you received as a child give you lifelong immunity. The half-life of that antibody is measured in the hundreds to thousands of years. And so it is possible for the immune system to develop a durable antibody response. We just need to capitalize on and understand the rules of the road. So both of those are going to require a lot of antigen, a lot of immunizations. There's one more obstacle on the B cell response that needs to be it's being worked on, but we need to overcome it. And that has to do with the immunogenicity of envelope. So that envelope protein of HIV is really poorly immunogenic. Some think it's because of it doesn't really engage naive B cells well. Some think that it has to do with the amount of surface glycosylation that HIV puts up, and those glycans look to the immune response like self. So they're called self-glycans. But whatever the reason, we need ways of overcoming that to get those broadly neutralizing antibodies. They're able to neutralize lots of different isolates. So that continues to be a major obstacle that we need to overcome as a field.
So you've talked about CD8 T cells a fair bit, and that the number in these patients was not diminished. And a number of recent studies have provided evidence for the role of these cells in restricting lentivirus replication in vivo. However, the reason the CD8 T cell response fails to control HIV remains incompletely understood. So can you expand on that and explain this work, please?
So as I mentioned earlier, we do understand that the people who are unable to control HIV have a hard time in their CD8 T cells proliferating, so they don't expand well, and they also don't increase the bullets of the immune system, so this molecule perforin that pokes holes in the target cell. The thing that we don't understand is what the mechanism of all that is at the subcellular level. And the reason that that's important is that worldwide, lots of groups are spending a huge amount of resources on immunotherapies, but yet they don't know what they're shooting against. Okay. And so they're giving cytokines and checkpoint blockade like anti-PD-1 and, all sorts of different molecules and vaccines, but yet we don't know at the subcellular level what the target should be. And we do have a pretty good idea that it is reversible, that you can make those cells then kill and upregulate perforin. But we don't know at the subcellular level exactly the mechanism of that. So that is going to be very important in the coming years to really understand, so that we can, once you know that, there are a lot of things out there that potentially have been developed for cancer or other applications that might be brought to the field that might work as really effective immunotherapies. And so that's where our lab is heading, and I think other labs as well.
So we've talked about, and to stay on this vaccine topic, because it's so fascinating, right? So your lab has examined the components of vaccine that contribute to an effective immune response to HIV, influenza, SARS-CoV, coronavirus 2. How do the immune responses to HIV compare with those other viruses like SARS, so on and so forth? Are there any insights that are cross-applicable, should we say?
There are, the immune response to HIV is peculiar in its the poor immunogenicity of the sort of makeup of HIV, the envelope and other factors that really lead to an ineffective immune response. Whereas, um, you know, nothing could be an easier target than SARS-CoV-2 spike. It makes all vaccines look wonderful, right? So you can, in the case of SARS-CoV-2, you can take a little piece of the receptor binding domain and put it into animals and get protection. Whereas for HIV, we've done these experiments. Actually, we're about to submit a paper where we made virus-like particles and we covered them in influenza H5. And then we made other virus particles that we covered in SARS-CoV-2 spike. And then the same example with HIV envelope. And you give it to experimental animals, H5, 2 shots, neutralizing antibodies in the thousands. SARS-CoV-2, immediate immune response, neutralizing antibodies in the thousands. You do that with HIV, nothing happens. So it is a formidable pathogen, but we know what we're looking for now, which is different than back in, the late 90s and 2000. And, we just have to get there.
So, a little bit just for my personal edification, has there been any, and forgive me for being so ignorant and not being up to date, but is there any further elucidation of the origin of HIV? I seem to remember there was a suggestion it was also from bushmeat, zoonosis that had jumped species.
Yeah, it looks, the great work has been done on this by a woman named Beatrice Hahn. But yes, it looks like it came from a specific population of animals in Africa through zoonosis. And so I can't remember all the details, but actually they have it refined down to the actual, sort of group of animals in the area. I forget if they were bonobos or other chimpanzees, but yes, it was a cross species
Yeah.
transmission.
I was talking to a gentleman, a scientist who's a virologist, who was telling me that in one species of South Chinese bat, there were thousands of virus species that looked like they had the capability to jump. And anyone who doesn't believe it, I've said this before. I was spent a fair bit of time in Queensland in Australia and visited over the years and seen with the growth of population, the rainforest, which did come down to the seafront, has all been replaced by houses. And at night, as dusk hits, bats in their gazillions are flying and defecating everywhere. And you just wonder, oh my goodness gracious, what have we done? So, Mark, I always like to finish with a bit of a fanciful question. I've already asked you about your favorite meal and your favorite wines. What about some wishes? A magical genie gives you 3 wishes for your area of healthcare. What would you wish for?
I think something that would last, through the millennia would be if vaccine immunology were developed as a field. Right now you have guys like me, we're sort of scattered around the world that look at this in a very intense way, but it hasn't coalesced into a field where we can agree on and make the rules that future generations would be able to say, oh, well, we just had a new outbreak of a new coronavirus. This is going to be the most effective way of combating that, that kind of thing. If this really coalesced into a discipline, I would like to see that. And I would like to see the sort of rules sort of emblazoned, in stone about how you fight this and what you're going to need to induce. And it may have similar ramifications for cancer and other chronic virus infections. I would also, my second wish would be to see a better model for the interaction of corporations and basic science. Right now, it's this very sort of scattered field where, government and philanthropy is developing some of these things, and then corporations come in. You might spend 12 years in an agreement with a corporation developing something, and then the company gets bought, or the business folks come in and say, I'm sorry, that's a wrap. We're not, even though there's tremendous promise, it frequently just gets shut down. And that really costs us as a field in development of immunotherapies and vaccines, because it's a huge amount of wasted effort. And similarly, it's all you needed to do was look at the list of vaccines back in March of 2020, the WHO list. And you would see that probably, if you asked any vaccinologist, about 90% of those 110 vaccines that were being developed probably had no business being there. They were just not going to work. And so, again, it's a lot of waste. Whereas if people were working together, they could de-risk for the corporations so that the government is funding vaccines and immunotherapies that clearly have a path to ultimately working or have a lot of promise. And so that companies could invest, but it wouldn't be a deal breaker for them to invest. And then they can take the ball for the last 10 yards or 5 yards, to the goal line. So I would like to see a better model for that. Right now, what's happening is we sort of exist in our own bubbles. Occasionally we bounce into each other at meetings, but that's sort of the sort of disorganized model for that. And then my last wish would be, just like probably everybody you would have on your show, is that science would be a greater priority for people. That you look at the success, you talk about vaccine hesitancy, look at the child mortality rate at the turn of the last century and the incredible advances that were made by antimicrobials and vaccines in the past 100 years. And then you look at the budget, and you just can't believe that this is not a higher priority for our people and for other governments worldwide. And so I would really, if I could wave a magic wand, that would be something that I would like to see, that this really needs to be a priority.
Yeah, fascinating. I think I'm going to need another conversation with you, Mark, because I've just come across a technology that looks at a simple finger prick to demonstrate antibody immunity to 11 vaccine preventable diseases. And they have some fascinating data on looking at people who have been vaccinated for things like measles and, so on and so forth, who are not demonstrating an immune response years after their vaccination. So maybe that could be a way to change people's understanding of their own risk. And I think there have been recent outbreaks of whooping cough and measles in the United States and elsewhere. So I think we're going to have you back on for further conversation. Mark, I found this absolutely fascinating. And I tell my students and junior colleagues that if you're going to present something, don't use complex words. Chances are you're presenting because you're the smartest person in your field in the room, right? But better to patronize than baffle. You have done a masterful job explaining to, a bit of an idiot savant. I've understood it, and I really appreciate you taking the time to do this. Very valuable, Dr. Mark Connors. Thank you, sir.
Oh, well, thank you, Jonathan. It's really been a pleasure to be with you, and thank you for the opportunity to talk about our work. And shoot me an email with what you decide to drink tonight with your meal.
Yeah, I'm still thinking about that, but absolutely, it's a bit, it's a spicy meal, so it's going to have to be something with some.
Go big.
Yeah, I'm thinking maybe even an Australian Shiraz. A bit crude. I'd like a Barolo, but I think it'll overwhelm it. But we'll talk some more, Mark, for sure. And folks, if you've enjoyed listening to this episode, please subscribe on Spotify, Apple, or wherever you get your podcasts so you never miss an episode. Like us, leave a review, tell your friends and colleagues. Check out the archives. There's hundreds of episodes with people every bit as fascinating as Dr. Mark Connors. And please tune in next week for another fascinating episode of the MJ podcast. Until then, I'm Dr. Jonathan Sackia. Thank you for listening. Please, everyone, stay safe, stay well, stay curious. Bye for now.